Welcome to the lecture of Plant Molecular Biology on plant tissue culture aseptic techniques. Plant tissue culture broadly refers to the in vitro cultivation of all plant parts under aseptic conditions on a nutrient culture medium of known composition. The excised piece of tissue or organ obtained from a plant to be cultured is called an explant. The explant can be taken from many different parts including portions of leaves, stems, roots, cotyledons, embryos, and many types of mature cells. However, the selection of an explant largely depends on the purpose of doing tissue culture, suitability of an explant for initiating cultures, and availability of an explant. While many different explants can be used for tissue culture, axillary buds and meristems are most commonly used, especially for micropropagation. Other desirable properties of explants include easily sterilizable, juvenile, and responsive to culture. The success of plant tissue culture depends on the choice of suitable growth medium. Tissue culture media should generally contain energy sources or macronutrients and inorganic salts or micronutrients to support plant cell growth. The media can be in the form of liquid or semi-solid, depends on the types of culture. Other requirements for plant tissue culture include plant growth regulators, biofertilizers, and vitamins. External parameters such as light, temperature, and pH, and subculturing frequency also play roles in successful tissue culture by supporting plant growth and tissue development. Aseptic technique is essential for the successful establishment and maintenance of plant cell tissue and organ cultures. The in vitro environment in which the plant material is grown is also ideal for the proliferations of microorganisms. In most cases, the microorganisms outgrow the plant tissues, resulting in their death. Contamination can also spread from culture to culture. The purpose of sterilization or aseptic technique is to eliminate the contaminants or minimize the possibility that microorganisms enter or remain in the cultures. The list of organisms described as contaminants in plant tissue culture includes bacteria, fungi, viruses, and so on. Contamination with bacteria is thought to be the most serious and has been described extensively in the literature. Bacteria are usually introduced with the explant and may survive surface sterilization if embedded in the interior tissues. Some bacteria spores can also survive the sterilization procedure even if they are on the tissue surface. Many kinds of bacteria have been found in plant tissue cultures, and they can be recognized by a characteristic odors with many different colors including white, cream, pink, and yellow. Fungi may enter cultures on explants as plant pathogens. They are frequently present in soil, or spores may be airborne. Fungi may be recognized by their fuzzy appearance and occur in a multitude of colors. Next contaminant is virus. Viruses are extremely small that are not easily detected. Thus, plant culture is not necessarily pathogen-free even if microorganisms are not detected, and viral contaminant can influence culture success. Contamination in tissue culture can originate from two sources, either through carryover of microorganisms on the surface or in the tissues of explants, or through faulty procedures in the laboratory and tissue culture technique. Most contamination is introduced with the explant because of inadequate sterilization or just very dirty material harvested from the field. Initial contamination is obvious within a few days after cultures are initiated. Latent contamination is usually by bacteria and is often observed long after cultures are initiated. Apparently, the bacteria are present endogenously in the initial plant material, but not obviously pathogenic in situ. Once in vitro, however, they increase in titer and overrun the cultures. Latent contamination is particularly dangerous because it can easily be transferred between cultures. Contamination can also occur as a result of poor sterile technique or dirty lab conditions. This kind of introduced contamination is largely preventable via proper care.